Welcome to the Deep Dive, where we provide the shortcut to being well-informed. Today, we aren't just reading a chapter. We're basically building an entire workshop.
That's a great way to put it.
We are systematically cracking open the foundational laboratory techniques that define modern cell and molecular biology. This is the toolkit, the stuff scientists use every single day to turn theories about life into observable, measurable facts.
That's right. Because if you want to understand how a cell actually works, you first need the experimental means to manipulate it and to observe it.
Exactly.
Our mission today is to walk through this toolkit sequentially, starting with the most basic problem in all of cell biology. How do you even see something that is vastly smaller than the tip of a human hair? We'll move step by step from overcoming the physical limits of light through isolating single components like proteins and DNA, all the way to, cutting, pasting, and silencing genes to figure out what they actually do.
It's a massive scope. I mean, this covers everything from visualizing structures that are fractions of a wavelength of light all the way to manipulating genetic code with just exquisite precision. So let's start where all cell biology truly begins.
Yeah.
Looking closer. When we talk about microscopy, the immediate thought for most people is just making things bigger. But we know that's not the critical factor. I mean, we can make a blurry photo of a microbe huge, but it doesn't really help us see any new detail.
Exactly. The real hero of the story isn't magnification, which is just the size increase. The real hero is resolution.
Right.
Resolution is defined as the ability to see two neighboring points in the visual field as two distinct, separate entities. If you just, crank the magnification dial beyond that point, the little light disks produced by those 2 points just overlap.
You just get a bigger blur.
You get a bigger blur. You can't resolve them. That resulting blur is what we call empty magnification. It's basically an optical...
An optical lie. You're not seeing more detail. You're just seeing a bigger fuzzy spot.
Precisely. An optical lie.
And what determines that fundamental limit? Why can't we just, make a better lens?
Because the limit is imposed by the wave nature of light itself. It's a phenomenon known as diffraction. To be resolved, an object needs to be larger than about half the wavelength of the light you're using to illuminate it.
So it's a physical law, not an engineering problem.
It's a hard law of physics.
Yeah.
And when you run the numbers for visible light, the practical diffraction limit of a conventional light microscope is slightly less than 0.2 micrometers or 200 nanometers.
That 200 nanometer limit, that really feels like the brick wall for traditional light microscopy.
It is the hard wall. Now, 200 nanometers is enough to resolve some of the larger organelles, like the nucleus or mitochondria, maybe a large vacuole in a plant cell.
The big stuff.
The big stuff. But the finest structures, the individual proteins, the ribosome machinery, the lipid bilayer itself, all of that is just fuzz. For comparison, the limit of resolution for the naked eye is about 0.1 millimeters, so we've come a long way, but we are still missing all of those crucial molecular details.
Okay, so before we even figure out how to break that 200 nanometer barrier, we have an even more basic problem. Cells are nearly invisible, they're mostly water, they're highly translucent, you look right through them, not at them.
And that brings us to the second key concept, visibility, or contrast. It's just the difference in appearance between an object and its background. The analogy here is simple and it's perfect. Stars.
Okay.
The night sky is filled with thousands of them, right? But they disappear entirely during the day, not because they went anywhere, but because they lack contrast against the much, much brighter sunlit background. In the lab, we have to create that contrast in the translucent cell.
And traditionally, the way to enhance contrast has been through staining, right? Just adding some color.
Correct. We use chemical dyes, or stains, that bind specifically to certain biological molecules, say DNA or protein, and they absorb certain wavelengths of light. This causes the object to appear colored and distinct from its background.
And there are specific stains for specific things.
Yes, a classic example from the book is the Fulgen stain. It's specific for DNA, so if you apply it to a tissue sample, say an onion root tip cell that's in metaphase, it causes the condensed chromosomes to appear vividly colored against a pale background. It's fantastic for fixed specimens.
But fixed means dead, and that's the big trade-off.
That is precisely the trade-off. Most stains are toxic, or the staining procedure itself requires these harsh, toxic conditions. The Fulgen stain, for example, requires acid hydrolysis. So if you want to watch a cell crawl or divide or respond to a hormone in real time, you just cannot use these traditional staining methods. #BB, techniques for unstained living cells.
Okay, so if we want a window into a cell that is actively living, we have to bypass chemical stains and use some kind of purely optical trick. This is where phase contrast microscopy comes in, allowing us to see these small, highly transparent, unstained specimens.
Phase contrast is. It's just ingenious because it exploits tiny inherent differences in the material itself. All the internal components of a cell, the nucleus, the cytoplasm, the organelles, they all have slightly different refractive indices.
Because they're made of different stuff.
Exactly. They contain different concentrations of proteins, DNA, RNA, water. These differences cause the light passing through them to slow down or speed up just a tiny bit, which shifts the light waves out of phase with each other.
And what the phase contrast microscope does is it translates that invisible phase shift into something our eyes can actually perceive.
Yes. It converts those really subtle differences in refractive index, which our eye normally cannot detect at all, into differences in intensity, which just means relative brightness and darkness.
So a transparent cell suddenly has texture.
Suddenly, a uniformly translucent cell shows all this internal structure, the nuclear envelope, the boundary of the mitochondria, the outlines of vesicles, all visible right through the eyepiece without ever adding a single drop of toxic stain. It's the standard solution for observing live, unstained tissue culture cells. #see fluorescence microscopy and live cell imaging.
Phase contrast is great for seeing the general outline and movement of the cell. But if you want to track a specific molecule, say a single type of receptor moving across the membrane, you need a label that's as precise as a GPS tracker. And that takes us into the molecular world of fluorescence.
And this is where modern cell biology truly exploded. Fluorescence relies on these special molecules called fluorophores or fluorochromes. What they do is they absorb energy from invisible short-wavelength light, typically ultraviolet or UV.
And then they spit it back out as visible light.
Almost instantly, yes. They re-emit a portion of that energy at a longer visible wavelength. So you shine blue light on it and it glows green.
So the resulting image in a fluorescence microscope must be inherently high contrast.
Oh, absolutely. Because the only light that's actually reaching the detector is the light re-emitted by this specifically labeled object. So those objects appear brightly colored, say neon green or brilliant red, against an almost entirely black background. It's perfect contrast.
But the real game changer here, the one that allowed us to move the labeling inside the living cell without any toxicity, was the discovery of the green fluorescent protein, or GFP.
This was a genuine scientific revolution. GFP was purified from the crystal jellyfish, Icoria victoria, and its gene was cloned in the early 1990s. The amazing thing about GFP, the real magic, is its mechanism. Its light-absorbing and emitting part, the chromophore, it forms through a process of self-modification.
It builds itself.
It builds itself, an autocatalytic reaction. This means it doesn't require any external enzymes or cofactors that might be missing in a different organism, like a mouse cell or a human cell.
Which opens the door to creating a chimeric protein.
Precisely. Using what's called recombinant DNA technology, researchers can take the gene for GFP and genetically fuse it to the gene for any protein they want to study.
So you're making a hybrid protein.
A hybrid protein. When the cell translates that fused gene, it produces a single, large, functioning protein that just happens to be fluorescently tagged. This lets us track the spatial and temporal distribution of that protein in living cells watching cell division, membrane trafficking, how a nucleus moves, all in dynamic real time, not just a fixed snapshot.
And the engineering didn't stop with just green. By mutating the GFP gene, researchers like Roger Tsien were able to generate an entire fluorescent palette.
Yes, variants like BFP, which is blue fluorescent protein, YFP for yellow, CFP for cyan, plus a whole family of related proteins from other organisms like DS red.
Having a whole palette means you can study the dynamics of multiple proteins at the same time, watching how they interact.
And the sources highlight just a stunning example of this complexity. A study observing competition between neurons in mice at their neuromuscular junctions. Researchers generated mice whose neurons contained different colored fluorescent proteins. Let's say some were cyan, others were yellow.
So you could literally see which neuron was which.
You could watch the branches from the different neurons compete directly for synaptic contact in real time in a living animal.
And what did that multicolor live imaging actually reveal?
The finding was striking and definitive. It turned out that all the winning synaptic branches belong to a single neuron, and all the losing branches belong to the other neuron. This visualization provided powerful, direct evidence for a mechanism of competitive exclusion in neuronal wiring, something that would have been completely impossible to observe in real time with any static staining method. #D, optical sectioning, and super resolution.
Okay, so fluorescence allows us to see specific molecules, but if the specimen is thick, say a whole embryo or a large tissue section, the light that's being emitted from above and below the plane you're trying to focus on, it just floods the detector and blurs the image. We need a way to filter out all that unwanted noise.
That technical challenge led directly to the development of laser scanning confocal fluorescence microscopy, or LSCFM. The whole concept is to illuminate and detect light from only a single, very thin plane, an optical section, at a time.
So how does LSCFM perform this optical sectioning? How does it slice the light?
The specimen is illuminated by an extremely fine, focused laser beam that rapidly scans across the target plane. But the crucial feature, the real trick, is the alignment of the components. The light that's emitted from the fluorophores in the focal plane passes through a tiny physical barrier. It's called the pinhole aperture.
And that's positioned very precisely.
It's positioned confocally with the illuminated spot.
So the pinhole is the gatekeeper.
It is the gatekeeper, exactly. Any light that originates from above or below the focal plane is physically displaced from that exact focus point, and so it's blocked by the pinhole aperture. It never even reaches the detector.
Which cleans up the image dramatically.
Dramatically. You can see this so vividly in the source materials, which show these clear micrographs of 3 separate optical sections, each maybe 0.3 micrometers thick, of a yeast nucleus. And since the sections are so clean, you can then merge them digitally to reconstruct a high-resolution, 3-dimensional model of the entire object.
Confocal imaging got us a clear 3D picture, but it didn't break that original 200 nanometer diffraction limit of light. If we want to see individual proteins clustering together, not just the organelles, we have to go even smaller. This is the realm of super-resolution microscopy.
This is truly modern biology, developed in the 2000s, and it's finally bypassing the limit imposed by the physics of light. The foundation lies in manipulating the behavior of the fluorophores themselves, specifically using engineered photoactivatable or photoswitchable proteins, which are often just mutants derived from the original GFP.
Tell us about one of the specific techniques that achieves that kind of precision.
Okay, let's look at STORM. That's stochastic optical reconstruction microscopy. The core trick is that instead of turning all the fluorophores on at once, which would just create that blurry 200 millimeter image, STORM cycles them on and off randomly.
So only a few are lit up at any one time.
At any given moment, only a tiny, statistically manageable subset of molecules is actually emitting light. And because they are so sparsely lit, the light from each individual molecule can be localized with extreme precision, far, far exceeding the diffraction limit.
So you're taking thousands of these high-precision single-molecule snapshots and then stitching them all together.
That's the genius of it. This process allows the system to localize single fluorescent molecules with a resolution below 20 nanometers. That's a 10-fold improvement over conventional light microscopy. The visual contrast is just stunning.
How so?
Well, if you compare a conventional fluorescence image where structures like microtubules and clathrin-coated pits appear as this sort of interconnected orange blur, the corresponding STORM image shows those same structures clearly separated and distinctly resolved. It's like moving from a pixelated photograph to an engineer's blueprint. It's a whole new level of detail.
Okay, so light has been pushed to its absolute physical maximum. Now, if we want to see cellular structure on the scale of angstroms or, low nanometers, we absolutely must switch from using light photons to using a beam of electrons. Tag A, transmission electron microscopy, TEM.
That's the big leap to electron microscopy. And we should probably first clarify the two major types. The transmission electron microscope, or TEM, acts like a slice viewer. It forms images using electrons that are transmitted through the specimen.
On the other one.
The scanning electron microscope, or SEM, on the other hand, forms images using electrons that bounce off the specimen's surface. That's what gives us that spectacular 3D topographic view of cell exteriors, like an insect's eye or a pollen grain.
The theoretical resolution of a TEM should be astounding, given the incredibly short wavelength of an accelerated electron, something like 0.05 angstroms. Why do we only practically achieve, say, 10 to 15 angstrom resolution for cellular structures?
That huge discrepancy is the enduring technological hurdle. It's primarily due to an unavoidable flaw called spherical aberration in the magnetic electron-focusing lenses. It forces us to use a very, very small numerical aperture, which essentially makes the lens less effective and drastically limits the practical resolution to those higher numbers.
In the physical machine itself, it sounds quite different from a light microscope.
Oh, it is imposing. The TEM is a tall, hollow, cylindrical column that has to operate under an extreme high vacuum. This is non-negotiable because any air molecules would just scatter the electron beam instantly.
Right, no error.
And a high voltage is applied to accelerate the electrons from a heated tungsten cathode, and magnetic coils serve as the lenses. Unlike a light microscope, the TEM's objective lens provides the entire range of magnification just by adjusting the current supplied to those magnetic coils.
Now, since biological material is mostly carbon, oxygen, and hydrogen, it has very low intrinsic power to scatter electrons. So if the electrons just pass right through without interacting, we get no contrast. How do scientists create the necessary contrast for a TEM?
We have to use heavy metals for staining. Things like osmium tetroxide or lead salts. These metals have high atomic numbers, which means they have high electron density. They penetrate the tissue and bind selectively to different structures. For example, osmium binds really strongly to the fatty acids in membranes.
So dark spots on the image mean more heavy metal.
Exactly. Regions that bind more heavy metal will scatter more electrons, meaning fewer electrons reach the fluorescent screen or the film below, and that causes that area to appear darker in the resulting image.
This sounds like the specimen preparation must be an absolute nightmare of precision.
It is incredibly complex and demanding. First, the tissue pieces must be minuscule, typically less than 1 millimeter cubed. Then the first critical step is fixation.
Freezing it in place.
Chemically freezing it, yes. This involves chemicals like glutaraldehyde, which rapidly cross-links all the proteins, and osmium tetroxide, which stabilizes and preserves cellular membranes.
Okay, but this fixation process immediately raises the concern of artifacts, right? You're chemically preserving the cell, which might create structures that weren't actually there in life.
That is the constant worry in electron microscopy. An artifact is a structure formed by, say, coagulation or precipitation during the fixation process that did not exist in the living cell. The best way to argue against an artifact is to demonstrate the same structure in unfixed cells, often using cryofixation, where you instantly freeze the tissue at liquid nitrogen temperatures. That avoids all the chemical cross-linking.
Okay, so once it's fixed, the sample needs to be cut. And since electrons can't penetrate very far, the slices have to be ridiculously thin.
Absurdly thin. After dehydration and embedding the tissue in a hard plastic or wax, we have to cut ultra-thin sections, about 100 nanometers thick, using a special device called an ultramicrotome. It's equipped with an exceptionally sharp diamond or glass knife.
100 nanometers, that's incredibly thin.
It is. These thin sections are then placed on a little mesh grid, stained with more heavy metals, and only then are they ready for viewing in the TEM.
You also mentioned a way to look inside a membrane structure without cutting the whole cell open, a way to see the topography of the lipid bilayer.
Right, that refers to freeze-fracture replication. Here, the tissue is rapidly frozen, and then it's struck with a blade, often under a vacuum. The fracture line has this tendency to propagate right along the middle, weakest plane of the lipid bilayer.
So it splits the membrane open.
It splits it right down the middle, exposing the internal faces and showing all the proteins embedded within.
And then you create a replica of that surface.
Correct. The exposed fractured surface is used as a template. A thin layer of a heavy metal, often platinum, is deposited on it, making a replica. Then the underlying biological material is dissolved away with acid. The resulting metal replica, which captures the membrane's internal topography, is what you actually view in the TEM. And they use a trick called shadow casting, depositing the metal at an angle to accentuate the height differences and give the image a three-dimensional feel. ### B atomic force microscopy, AFM.
Okay, moving away from high vacuum electron beams, there are other methods of physical probing that offer extraordinary resolution while also being able to operate under normal conditions, like atomic force microscopy or AFM. How does AFM physically map a molecule?
AFM is radically different. It doesn't use electrons or photons. It uses mechanical force. It's like a tiny record player needle.
A very, very tiny needle.
An atomically sharp needle. The instrument scans a sharp, micro-sized tip that's attached to a flexible, oscillating beam called a cantilever over the specimen's surface. As the tip encounters variations in topography, even tiny atomic bumps and valleys, the cantilever's oscillation frequency changes due to atomic forces between the tip and the sample.
And those frequency changes are the data points.
Exactly. A laser is bounced off the back of that oscillating cantilever, and those tiny movements are tracked and mapped digitally to create a detailed, three-dimensional topographic image.
What makes AFM such a uniquely valuable tool compared to, say, X-ray crystallography or even cryo-EM?
Well, AFM offers two critical advantages that solve some major biological limitations. First, unlike those other techniques, which are averaging techniques that create a composite structure from millions of identical molecules, AFM provides an image of each individual molecule as it's oriented. It lets you see heterogeneity.
And the second advantage.
Second, and this is perhaps the most exciting part, is the development of high-speed AFM, or HSAFM. This allows researchers to observe dynamic processes in real time. We can literally watch a motor protein walking along a filament or an enzyme changing shape as it binds to a substrate. It overcomes the static snapshot limitation of almost every other structural method. You're watching biology happen.
So we've mastered seeing the cell all the way down to the molecular level. But what if we want to study the internal machinery, the specific proteins, the DNA, or the organelles outside of the complex, noisy context of the whole cell? This requires isolation, tracing, and analysis. #A, radioisotopes in research.
One of the foundational techniques for this relies on tracing molecules through metabolic pathways using radioisotopes. An isotope just means that atoms of the same element have the same number of protons but a different number of neutrons. For example, common hydrogen exists alongside its radioactive cousin, tritium, $3, which has 2 extra neutrons.
We use them because they are chemically identical to the stable version, so the cell handles them normally, but they're unstable and they decay, releasing detectable radiation. How is that instability measured?
It's measured by the half-life. This is just the time required for half of the radioactive atoms in a sample to decay. And the sources give a fascinating historical example. Early photosynthesis research had to rely on carbon-11, which has a half-life of only 20 minutes.
20 minutes, so they had to work fast.
The researchers had to synthesize the labeled molecules, feed them to the plants, run the entire experiment, and analyze the results in a matter of minutes. The text notes that these experiments were literally carried out on the run.
Wow. So the later availability of carbon-14C, with its half-life of 5,700 years, must have been a total game-changer for stability and experimental design.
Oh, absolutely. It changed everything. Now, the 3 main forms of radiation released are alpha particles, beta particles, which are equivalent to a high-energy electron and are very common in cell biology, and gamma radiation, which is electromagnetic radiation. We track the most commonly used beta emitters using detectors like liquid scintillation spectrometry or through autoradiography.
And what's that?
That's where you place the sample near a radiation-sensitive emulsion, like photographic film, and the resulting dark spots on the film reveal precisely where the isotope accumulated in the tissue. #B cell culture.
To get the large, uniform quantities of a single cell type that we need for molecular analysis, we rely very heavily on cell culture. This provides a simplified, controlled in vitro system.
Cell culture allows us to study all these complex, essential biological activities: cell division, movement, signaling, response to hormones or drugs, in isolation from the complexity of the whole organism. Historically, cell growth required these complex, natural fluids like animal serum, but modern labs are increasingly using complex, chemically defined, serum-free media, aiming for total control over the cell's environment.
And how do we initiate and maintain these cell lines?
Well, cells that are derived directly from an organism, say from an embryo or a tissue biopsy, form what we call a primary culture. Getting these cells often requires using proteolytic enzymes like trypsin and calcium chelators like EDTA to dissociate the tissue into individual cells.
And a secondary culture.
Any subsequent culture that started from a frozen stock or from a previous passage is called a secondary culture.
Now, the physical environment matters immensely here, which brings us to this conceptual distinction between 2-dimensional and 3-dimensional culture systems.
This is a really crucial point for understanding how in vitro results translate to what actually happens in the body. Most traditional studies use 2D culture, where cells are grown on a hard, flat plastic substratum. The source material notes that human fibroblasts, for instance, assume a highly unnatural, flat, pancake-like morphology in 2D.
Right, they're not built to live on plastic.
Exactly. In contrast, 3D culture, where cells are grown within a soft, supportive matrix, provides a far more natural environment.
What does that 3D environment reveal that the 2D doesn't?
Well, in a 3D culture, those same fibroblasts assume their natural spindle-shaped structure, which is much closer to their in vivo organization.
Yeah.
And this more natural morphology leads to major differences in signaling activities, gene expression, and cytoskeletal organization. It emphasizes that the geometry of the culture system is not just some technical detail. It fundamentally impacts cell behavior. #C, protein isolation and purification.
Okay, so once we have our millions of cultured cells, we need to purify the specific protein we care about from the thousands of others that are present in the cell homogenate. We often start by exploiting basic solubility differences, right?
That's the initial step, yes. It's called selective precipitation, and it's designed to remove the major contaminants early on. But for high-resolution separation, we have to turn to chromatography. That's the general term for fractionating a complex mixture, as its components are differentially retarded while moving through some kind of immobile matrix.
Let's look at two of the key techniques, starting with gel filtration chromatography.
Gel filtration separates proteins based primarily on their effective size, or what's called their hydrodynamic radius. The mixture is passed through a column that's packed with porous beads, usually made of a cross-linked polysaccharide like Cefidex.
And explain how those tiny pores in the beads do the separation work.
It's a trick that's based on path length. The mechanism ensures that the larger proteins are excluded from the internal aqueous spaces of the beads. They can't fit inside, so they're forced to follow a shorter, quicker path through the column around the beads.
So they come out first.
They elute first. The smaller proteins, on the other hand, can enter the pores, making their path much, much longer. So they are retarded and they elute later. If you were trying to purify a 125 kilodalton protein that was contaminated by a 250 kilodalton protein, the 250 kilodalton protein would exit first, followed by your 125 kilodalton target, and finally the 75 kilodalton contaminant would come out last.
That relies on size. But the most powerful technique, which relies on a specific molecular interaction, is affinity chromatography.
This is the purification powerhouse. It often leads to near total purification in a single step. It exploits the unique structural properties of specific binding, for example, a receptor binding to its ligand, or an enzyme binding its substrate, or an antibody binding its antigen.
And how is the column engineered for that kind of specificity?
The specific interacting molecule, the ligand or the substrate, is covalently linked to the inert matrix of the column. So when the crude protein mixture passes through, only the target protein, the one that specifically recognizes and binds to that ligand, is held back. All the other contaminating proteins just pass right through.
And then you just have to get your protein off the column.
Right. The target protein is then displaced or eluted, usually by changing the buffer conditions, like altering the ionic composition or the pH to disrupt that specific binding interaction. #TED protein electrophoresis and analysis.
Okay, so once we have a purified protein, or even if we just want to analyze a complex mixture, we need a high-resolution separation method. And that leads us to electrophoresis, specifically polyacrylamide gel electrophoresis, or PAGE.
PAGE is the general technique where we use an electric field to drive proteins through a molecular sieve made of polyacrylamide. But the most common and by far the most informative application is SDS-PAGE.
Sodium dodecyl sulfate, or SDS, is the detergent that's critical to this method. Why do we need it? What does it do?
SDS is the great equalizer. It binds proportionally to the protein's mass, about 1.4 grams of SDS per gram of protein, and it achieves two fundamental changes. First, it completely unfolds all the proteins into a similar rod-like shape, so it eliminates shape differences as a factor in migration.
And second,
It confers an equivalent high negative charge density to every single protein species in the sample.
so once shape and charge are made uniform across the board, what is the only remaining variable that's affecting the separation?
Molecular mass. That's it. Since the charge-to-mass ratio is essentially the same for every protein in the sample, the separation is based solely on size. Larger proteins are retarded more by the gel mesh, and so they migrate slower. By comparing the position of an unknown band to standardized proteins of known molecular mass, we can accurately determine the mass of our target protein.
Now, if you need to separate thousands of proteins simultaneously, maybe you're doing a comprehensive survey of all the proteins expressed in a particular cell type, you need the extreme power of two-dimensional gel electrophoresis.
That's the highest resolution separation available. It's capable of distinguishing most of the thousands of different polypeptides in a typical cell. It separates proteins based on two distinct properties, which are run in orthogonal dimensions.
And what are those two dimensions?
The first dimension is isoelectric focusing. This separates proteins based on their isoelectric point. That's the pH at which the protein carries no net electrical charge. Once they're separated across a narrow pH gradient strip, That strip is placed horizontally on a larger gel slab, and then the second dimension is run, SDS-PAGE, which separates those same proteins based on their molecular mass.
So you get a map of spots.
You get a unique map of separated spots, and it's ideal for detecting really subtle changes in protein expression or post-translational modifications like phosphorylation across different experimental conditions.
Finally, let's just quickly look at the analysis tools that are used once the proteins are purified or separated.
The most basic instrument is the spectrophotometer, which measures the amount of light that's absorbed by a solution in a quartz cuvette. This is useful for rapidly determining the concentration of nucleic acid or protein. But for definitive identification, we have to turn to mass spectrometry.
That seems like a very complicated piece of equipment. What does mass spectrometry fundamentally measure?
It measures the mass-to-charge, MZ ratio. The instrument converts molecules into charged gaseous ions, and then it separates those ions using a magnetic field. We can take a purified protein, fragment it into smaller peptides, determine the mass of each of those fragments with incredible precision, and then we input that unique set of masses into a genomic database.
And the computer does the matching.
The computer then searches for a protein sequence whose theoretical peptide fragments would match the observed masses, allowing for highly accurate identification.
We've covered how we see the cell and how we isolate and analyze its protein machinery.
Now let's pivot to the blueprint itself. The nucleic acids. A lot of the physical separation techniques share similar principles with protein analysis, but the specific tools are, of course, tailored for DNA and RNA. #A, nucleic acid electrophoresis, and centrifugation.
For separating DNA and RNA fragments, we still use gel electrophoresis, though we often use agarose gels instead of polyacrylamide for larger pieces. And since DNA is uniformly negatively charged because of its phosphate backbone, its rate of migration is governed almost entirely by size.
And how do we see the DNA?
The bands are visualized by staining the gel with ethidium bromide. This chemical intercalates or slips into the DNA helix and causes the bands to fluoresce intensely under UV light. The method is incredibly sensitive. You can separate molecules that differ by as little as a single nucleotide in length.
Besides electrophoresis, we have ultracentrifugation, which is an indispensable tool for separating large molecules like DNA based on their physical properties. This is more about force than sieving. Correct. And there are two critical types of ultracentrifugation. First, there's velocity sedimentation. This uses a sucrose density gradient, and it separates molecules primarily by their size and shape. Larger, heavier molecules will move faster through the gradient in the powerful centrifugal field, hence the name velocity measure.
The second type separates based on intrinsic molecular characteristics, which sounds a lot more powerful.
That's equilibrium centrifugation, also called isopycnic centrifugation. It separates molecules strictly based on their buoyant density. High-speed, long-term centrifugation establishes a stable gradient of a heavy metal salt, like cesium chloride.
So the 2 itself is a density gradient.
It has a stable density gradient from top to bottom. The DNA molecules then migrate through that gradient until they reach the precise position where their own buoyant density matches the density of the surrounding medium. And there they form a sharp band.
And this technique is sensitive enough to differentiate between molecules that are only subtly different from each other.
It is extremely sensitive. It can separate DNA molecules with different base compositions. For instance, AT-rich DNA is less dense than GC-rich DNA, or even molecules containing different isotopes. This was the method used in the famous experiment that used DNA containing the heavy isotope nitrogen-15 versus the light isotope nitrogen-14 to prove how DNA replicates.
We also use nucleic acid hybridization to understand not just molecular identity, but also evolutionary relationships.
Right. Hybridization measures the similarity in nucleotide sequences between two different DNA or RNA samples. You can take DNA from two distant species, separate the strands, mix them together, and let them reanneal. If the species are distant, the resulting hybrid duplexes will have significant base pair mismatches.
Which makes them less stable.
Less stable. And this instability is measurable as a lower melting temperature, or $T$. This provides a direct, quantifiable gauge of the evolutionary distance between the two organisms. ###SHAB-B, recombinant DNA technology and cloning.
Okay, now we enter the core of the molecular toolkit, starting with how we create recombinant DNA. That is, molecules containing DNA derived from more than one source. This entire field was launched by the discovery of restriction endonucleases.
These are the molecular scissors, and they are astonishingly specific. Restriction enzymes recognize and cleave specific symmetric nucleotide sequences. The classic example is ECOR1, which recognizes the sequence GAATC. And crucially, they often leave these sticky ends, short, single-stranded overhangs that are complementary.
And this complementarity is what allows us to combine DNA from pretty much any source.
Exactly. By cleaving DNA from, say, a human cell and a bacterial plasmid with the same restriction enzyme, you generate fragments that have complementary sticky ends. These can then be ligated together to form a recombinant molecule. It's essentially cutting and pasting DNA.
And the purpose of DNA cloning is to produce massive quantities of a specific DNA segment, say, the gene for human growth hormone.
To do that, the segment is linked to a vector DNA. This is typically a small circular piece of bacterial DNA called a plasmid, or sometimes the genome of a virus like phage lambda. The vector acts as a vehicle for replication. This recombinant molecule is then introduced into a host cell, usually E. coli.
And the bacteria do all the work.
The bacteria do the work. As the host bacteria grow and divide, they replicate that foreign DNA segment exponentially, producing millions and millions of copies. A clone.
So once we have millions of these bacterial colonies, each potentially containing a different piece of foreign DNA, how do we find the one that contains the specific gene we actually want?
We need a rigorous screening process. First, the bacterial cells are plated out to form separate colonies or clones. Then we use a technique called replica plating to create identical copies of those colonies on a separate membrane. One of these replicas is then subjected to in situ hybridization.
Using a probe.
Using a small known sequence of DNA, a labeled probe, that is complementary to the target gene, like the insulin gene, the probe will only hybridize to the DNA of the colony that contains that target sequence, allowing researchers to locate and isolate that specific clone from the original plate. ## Polymerase Chain Reaction, PCR.
Cloning is great for large-scale preparation, but if we just need to quickly amplify a trace amount of DNA, perhaps from a single hair follicle at a crime scene or a single cell, we use the invention that totally revolutionized rapid diagnostics, PCR, the polymerase chain reaction.
PCR revolutionized molecular biology because it amplifies a specific target DNA sequence in vitro in a tube without needing a host cell at all. The process relies on a clever evolutionary trick, the use of Taq DNA polymerase.
From a heat-loving bacteria.
An enzyme isolated from a thermophilic bacterium. This enzyme is highly heat-resistant, which enables it to survive the very high temperatures needed to separate the DNA strands in each cycle.
So walk us through that exponential amplification cycle.
The cycle has 3 basic steps, and it's repeated maybe 25 to 40 times. Step 1 is denaturation. The mixture is heated, usually above 90 degrees Celsius, to separate the double-stranded DNA templates.
Step 2.
Step 2 is annealing. The temperature is cooled to allow 2 synthetic primers, these are short oligonucleotides, to specifically hybridize or anneal only to the sequences that are flanking the target region.
And then the enzyme gets to work.
Step 3 is extension. The heat-stable Taq polymerase synthesizes a new complementary strand, starting from those primers. And because the products of one cycle become the templates for the next cycle, the amount of target DNA doubles each time. It's exponential amplification, #D DNA sequencing, Sanger-Dioxy method.
To really understand the function of any DNA segment, we have to determine its precise nucleotide sequence. This foundational knowledge was made accessible by the Sanger-Dioxy method.
The Sanger sequencing technique is beautifully simple in concept. It involves synthesizing new DNA strands in vitro, but in the presence of a low concentration of a chemically modified precursor, the dideoxyribonucleoside triphosphate, or DDNTP.
What's so special about the DDNTP?
It lacks the essential 3' hydroxyl group, the 3' OH group, that's necessary for the DNA polymerase to add the next nucleotide. So when a DDNTP is randomly incorporated into the growing strand, DNA synthesis terminates at that exact position.
So by including all 4 bases, A, T, C, and G of these terminated molecules, you generate this nested set of fragments, all starting at the same point, but ending at every possible location of that specific base.
Exactly. And to read the resulting sequence, each of the 4 DDNTPs is tagged with a unique fluorescent dye. The terminated fragments are then separated by high-resolution gel electrophoresis, typically in tiny capillary tubes. A laser detector reads the color of the band as the fragments pass by, and that reveals the identity of the terminal nucleotide. This sequence data is output as an electropherogram, which the computer then reads to determine the original sequence.
That system defined genomics for decades, but new technology has now superseded it, focusing on speed and cost.
That's the whole realm of third-generation sequencing. These newer instruments have shifted the focus towards sequencing single DNA molecules. This eliminates the need for preamplification and allows for much, much longer reads, operating faster and cheaper. A prominent example involves using nanopores,
Tiny holes.
tiny holes through which a single DNA molecule is pulled. As each individual nucleotide passes through the pore, it disrupts an ionic current in a characteristic way, allowing the system to identify the sequence in real time.
We now have the ability to read the code, replicate it, and isolate components. The final major section of this experimental toolkit focuses on how we take that knowledge and use it to actively manipulate the cell, often by altering or limiting a gene's function to understand its biological role. #TAG-HAGDR DNA transfer, transgenics.
When we introduce foreign genes into a multicellular organism, we're creating a transgenic animal. For mice, the gold standard method involves the direct microinjection of the desired DNA right into the nucleus of a fertilized embryo.
And the goal is for it to stick.
The goal is for that foreign DNA, the transgene, to integrate stably into the host chromosomes. If that's successful, the transgene is then passed on to every subsequent cell, and crucially, it's inherited by the offspring. This allows researchers to study the function of a human gene, for example, in the context of a developing mammal.
And plants are often modified using a bacteria known for causing tumors, which is a neat twist of biology.
Yes, transgenic plants are often generated using the tDNA transformation system. It's derived from the soil bacterium Agrobacterium tumefaciens. This bacterium naturally contains something called the T plasmid, which carries a segment of transfer DNA, or tDNA, that the bacterium normally transfers into the host plant cell's chromosomes, and that's what causes tumorous growth.
So scientists just hijack that natural transfer system.
Precisely. Scientists splice the foreign genes they want introduced into the agrobacterium's modified T plasmid. When the bacteria then infect plant cells, they transfer the engineered recombinant tDNA containing the foreign gene into the plant cell nucleus. The transformed cells, often meristem cells at the shoot apex, can then be rooted and grown into mature plants. This is the technology used to introduce traits like insect resistance via insect-killing toxins. #B, determining gene function, reverse genetics.
For the vast majority of genes that were identified in all the genome sequencing projects, we know the sequence, but we have no idea what they do. This necessitates reverse genetics.
Reverse genetics is the approach of starting with a known sequence, the genotype, and actively eliminating its function to determine the resulting change, or the phenotype. This is the opposite of forward genetics, which starts with a mutant phenotype and then works backward to identify the responsible gene.
One way to study small changes is in vitro mutagenesis.
That allows for exquisite control. A cloned gene segment is mutated in a predetermined way, say, changing a single base pair to substitute one specific amino acid for another, to study how that subtle change affects the resulting protein's function. Maybe it alters its binding capacity or its enzymatic rate.
For whole organisms, the gold standard has historically been the knockout mouse.
Right. Generating a mouse that's lacking a functional gene or a knockout is a lengthy, laborious process involving techniques like homologous recombination. However, the complete absence of many essential genes is often lethal to the embryo.
Well, you can't study it.
It prevents us from studying its function in adult tissue. So to bypass this, researchers developed conditional knockouts. This uses specialized genetics to allow the researchers to switch off a specific gene only in desired tissues or at desired times later in life, making adult function analysis possible.
But the fastest, cheapest, and most widespread method for eliminating gene function now is RNA interference, or RNAi.
RNAi is a widespread natural cellular process that we have harnessed for our own purposes. It utilizes these small 21 nucleotide double-stranded RNA molecules called siRNAs, or small interfering RNAs. When you introduce these into a cell, the siRNAs are incorporated into a complex that specifically recognizes and induces the destruction of target messenger RNAs that have the complementary sequence.
So you're essentially turning the gene off at the message level without permanently changing the DNA.
Exactly. By degrading the specific messenger RNA, you prevent the protein from ever being made, effectively knocking down the gene's function quickly and cheaply. And the application is huge. The sources highlight large-scale screening. Researchers treated cultured Drosophila cells with over 14,000 different siRNAs and screened them to identify previously unknown genes involved in complex cellular processes like mitotic spindle assembly.
What's an example?
Well, the knockdown of a protein called aurora kinase using a specific RNAi caused severe defects in chromosome segregation, which definitively linked that gene to that critical function. Hashtag C, the use of antibodies.
Finally, we have to cover the indispensable tool that is the antibody. They are the ultimate molecular probes.
Antibodies, or immunoglobulins, possess a truly remarkable specificity. They are capable of distinguishing between two polypeptides that differ by only a single amino acid substitution. This makes them perfect molecular spies, able to identify a target protein out of a complex mixture of thousands.
And we distinguish between two main types of antibody preparations, starting with the less refined one.
That would be polyclonal antibodies. They're derived from the antiserum of an immunized animal. This serum contains a whole variety of different antibody species. They all bind to the same antigen, but they're targeting different parts of that antigen. Because they're structurally diverse, it's impossible to purify a single, uniform antibody species from this kind of preparation.
Which is why the development of monoclonal antibodies was another major revolution in the lab.
Monoclonal antibodies are produced by hybridomas. These are hybrid cells formed by fusing a normal, short-lived antibody-producing lymphocyte with a malignant, immortal myeloma cell. The resulting hybridoma is immortal and can be cultured in vitro indefinitely to produce large, virtually unlimited quantities of a single, highly specific antibody species that targets only one site on the antigen.
And these highly specific molecular probes are just central to almost every modern technique.
Oh, they are used across the board. For protein purification, where the antibody can selectively precipitate the target protein from a crude mixture. For identification, using techniques like the Western blot to confirm the presence and size of a protein. And for immunofluorescence, where antibodies are conjugated with fluorescent molecules to precisely localize antigens within cells or tissues using fluorescence microscopy, providing that crucial spatial context. Hashtag tag outro.
What an extensive tour of the experimental landscape. We started with the very real physical limits of light, learning how the phase contrast microscope uses refractive index to visualize living cells without any toxicity, and how the Discovery GFP transformed light microscopy into a tool for dynamic, live cell molecular imaging.
We then saw how technology fought back against physics, either by using the confocal microscope's pinhole to achieve those clean optical sections, or by employing storm and photoswitchable fluorophores to finally break the fundamental 200 nanometer diffraction limit. We stepped down even further to the nanometer scale with the TEM, relying on heavy metal staining and complex fixation methods, and the AFM to actually visualize single molecules dynamically.
On the component isolation side, we saw the power of chemical affinity, how gel filtration separates by size, and how affinity chromatography uses specific binding to purify target proteins in one powerful step. Then we use SDS-PAGE to resolve components purely by mass, followed by mass spectrometry for that definitive identification and sequencing.
Finally, we covered the core molecular manipulation techniques: restriction endonucleases as our molecular scissors, DNA cloning for amplification in vectors, the extreme exponential power of PCR for in vitro amplification, and the role of ddNTPs in Sanger sequencing. And crucially, how RNAi provides a fast, cheap method for reverse genetics knocking down gene function to figure out its resulting phenotype.
This entire toolkit, from the century-old concept of resolution to modern single-molecule sequencing, it really reinforces that these methods are not merely background noise. They are the engine of discovery. They provide the definitive, tangible evidence for every structure and function we understand deep within the cell.
Indeed. It is through the consistent application and the evolution of these rigorous experimental methods rooted in basic physics and chemistry that cell biology as a discipline continues to push the boundaries of what we know about life.
Thank you for joining us on this deep dive into the experimental toolkit of cell biology.